Our next talk is going to be from Dr. Amlan Chakraborty from Monash University in Australia, who's going to be discussing Serelaxin-nanoparticles abrogates inflammation and remodeling associated with chronic allergic airway disease through specific myeloid cell subsets. Perfect, we can see your screen, so go ahead when you feel ready.
Thank you so much. It's a nice and wonderful evening here out in Melbourne, down here south. And we had a lovely evening today with the Australian Open final and Novak Djokovic winning it. So welcome everyone. So today I'm going to talk about serelaxin nanoparticles abrogating inflammation in a chronic AAD or asthma setting. So globally, 4.3% of the population, that's about 339 million people, suffer from asthma. And it's a disease which is clinically diagnosed with constriction of the airways and developing of mucus in these airways, resulting in chronic exacerbations with repeated episodes of wheezing. So the therapies that are associated points towards the use of corticosteroids, but a certain population of people, around like 10% of these population, so are resistant to these treatments of corticosteroids, which increases the healthcare cost by 80%. And it's much more prevalent in children from 0 to 14 years, and that's also a grave situation. But what's much more concerning is about the airway remodeling side of asthma. So if you want to know about the pathophysiology of asthma, so it's a chronic inflammation where we have infiltration of eosinophils, T lymphocytes, macrophages, and there's goblet cell metaplasia, there's smooth muscle hypertrophy, epithelium shedding, but there's no affecting the lung parenchyma. But towards the airway remodeling side, so we are looking into subepithelial fibrosis as well as the development of airway smooth muscles. As you see on my panels on my top, so the alpha SMA, so that's in the OVA albumin asthma model, which I'm going to discuss in a while. So there has been the development of alpha smooth muscle actin, and there's been collagen deposition underneath the airways, which results in airway remodeling and thickening of the airway epithelium. So our lab investigates into developing different other therapies. So we look into serelaxin, which is the human 2-relaxin gene product, and it has been shown to have cardioprotective as well as antifibrotic effects. But this human serelaxin, which is a 6-kilodalton peptide, has a very short half-life. So we conjugated them into biodegradable super paramagnetic iron oxide nanoparticles, and we put them into a model of ovalbumin-induced asthma. So we sensitized mice with ovalbumin and then nebulized 3 times a week for 6 weeks with ovalbumin, and then treated mice with this serelaxin conjugated nanoparticles as a point of therapy to see if there's regression in airway remodeling as well as the inflammation side of asthma. So to our notice, what we found was that using these serelaxin nanoparticles, it definitely regressed the inflammation that is associated with asthma, as well as the goblet cell metaplasia. The number of goblet cells significantly reduced in this disease. Also, when we looked into the airway epithelial thickness, there was a certain reduction in the epithelial thickness that has been observed in the oval model. Also, there has been reduction in collagen deposition underneath the airways. So this also points out to the fact that of epithelial shedding, or like the damage in the epithelium, as pointed out by the development of TSLP protein. So we found that using these serelaxin nanoparticles, there has been a decrease in the presence of serelaxin protein. So looking into the inflammatory side, we started to investigate the cytokines which are involved. So we found the pro-inflammatory cytokines IL-1 beta, IL-6, and TNF has been reduced by serelaxin nanoparticles. And we found that also in a wide data set, observing different other cytokines as well as pro-inflammatory mediators and chemokines, the CCL2 and CCL11, 2 of these chemokines were highly active in case of the oval model. So the CCL2 is active to bring in influx of alveolar macrophages in the lung, and the CCL11 for the eosinophils. As we all know, one subset of asthma is eosinophilic. But to this note, it hasn't been discovered or hasn't been found yet the role of alveolar macrophages to a greater extent, or the subtypes of alveolar macrophages which are involved in this asthma. So we also resorted to look into those subsets, but before that, it is a disease of the airways, and has the airway remodeling sites. We investigated the effect of the profibrotic cytokine TGF-beta. So we found that TGF-beta onto the tissues has been reduced significantly with the nanoparticle relaxin. And therefore, we started investigating to the airway mechanics. So we measured the lung function. That is a way to measure airway hyperresponsiveness. And we found that the increased resistance due to asthma has been quite significantly reduced with the use of Serelaxin-nanoparticles and improved the elasticity, that's the dynamic lung compliance with the use of Serelaxin-nanoparticles, which kind of shows that it has a good translative effect if we can move this to the clinic. So talking about alveolar macrophages, we did a basic study to see whether these nanoparticles are actually taken up by macrophages or eosinophils. So we found that these nanoparticles of serelaxin are only taken up by alveolar macrophages in the lung in mice and not by eosinophils, which points us to the next part of the study to look into them. So we found that these nanoparticles, by coating them with FITC, are only localized in the cytoplasm and not in the nucleus. So they are non-inflammatory, as previous results shows that if nanoparticles penetrate the nucleus, they cause an inflammatory cascade. So moving on to the specific alveolar macrophages, which is the interesting bit of the talk, of like what is happening in this OVA model and which kind of macrophages are taking up. So to a greater extent, CD45 positive, F480, CD206 double positive macrophages, which I'm categorizing as an M2 skewed phenotype, are the major players here which are taking up these nanoparticles. Also, if we look into the confirmation that these are actually the alveolar macs, so we are looking into CD11C and CD206 positive macrophages, and it is the involvement of the CD206 population which takes up these nanoparticles, skewing towards the M2 phenotype. So we did some staining onto the lung sections of the OVA versus saline, and the classical INOS is a M1 marker, and we found that there has been almost negligible amount of M1 macrophages, but significantly higher amount of M2 macrophages in the OVA model. So focusing on my next part of the talk about the different macrophages. So along with the macrophage population, there has been a small number of about 17% of CD11C positive dendritic cells, which are also involved, as these are also the antigen presenting cells that you can see in the lung. But the presence of the marker CD11C is highly essential for the uptake, as nanoparticles like of this size and having a peptide conjugation are only taken up by antigen presenting cells having this CD11C marker. So looking into other macrophage population, the CD68 high tissue resident macrophages are the major players again. So we found that almost 7% of the CD68 population, which are the CD68 high expressing population, had the CD11C positive marker and also the F480 positive marker. And these are the major macrophages which are tissue resident and is responsible for the uptake of the Serelaxin nanoparticles. To the lesser extent, 19% of the population was CD68 low, but again, the presence of the CD11C marker was important for the uptake of Serelaxin nanoparticles. So then we started to look after whether this can be actually translated to human therapy. So with our collaborators in the Alfred Hospital, so we collected human lung fibroblasts from asthma and non-asthma patients and seeded them into collagen gels and stimulated them with the profibrotic factor TGF-beta to see whether we can actually regress fibrosis as well in human patients. So to a greater extent, Serelaxin nanoparticles could reduce the collagen, so prevent the reduction in the collagen gel size in both the asthma and the non-asthma patients, but was quite prevalent on the asthma patients, which is to a greater extent shows the clinical translatability of this work. So in summary, these Serelaxin nanoparticles are specifically taken up by alveolar macrophages, which sometimes get skewed towards the M2 phenotype in abrogating the chronic AAD that is observed, as well as tissue resident CD68 high macrophages. They take up 13% more of these nanoparticles, reducing the clinical features of airway resistance, the lung compliance improvement, reduction in pro-inflammatory cytokines, and significantly reducing airway inflammation, epithelial shedding and damage, as well as the only therapy that is showing to work towards remodeling associated with chronic AAD. So I would like to extend my thanks to the organizers as well as to my collaborators in University of New South Wales, Monash University, University of Melbourne, to the Monash Histology platform, the electron microscopy platform, flow cytometry platform, and of course, my research funding support from the Australian Research Council. And thank you so much for such a lovely conference. Thank you.
Perfect. Thank you so much, Dr. Chakraborty. That was a really great talk. I'm just going to open up now for questions. We have some questions in the Q&A, and if anyone has any other questions, feel free to send those questions through there. So the first question we have is, what is the toxicity level of Serelaxin-nanoparticles as it is used for asthma, and toxicity level can be the major issue?
So Serelaxin is a human hormone, and it is already present in the body in pregnant females, and the levels when we have like a triplet or a quadruplet pregnancy, it is the same level of the hormone that we are trying to use. So significantly, we have seen at least like 100 nanograms per ml concentration without affecting any cellular cytotoxicity. So the concentration at which we are using is 25 nanograms per ml. That is almost 1/4. So we haven't noticed any cytotoxicity so far.
Perfect, that's great. Thanks very much. The second question we have in the Q&A is, and how are, or I guess it's a follow-up of that first question, but how are Serelaxin nanoparticles synthesized, biological, physical, or chemical?
Yeah, so I would like to actually take you to one of my slides. So Serelaxin nanoparticles, so we use a EDC chemistry, so that's basically a peptide-based chemistry. So we synthesize superparamagnetic iron oxide nanoparticles and then coat them with carboxyl groups by citric acid. Then we use a EDC, a carbonamide reaction to conjugate this serelaxin onto the particles, as you can see through like a peptide bond. And to measure the levels of serelaxin that has been conjugated, so we use a relaxin ELISA, and it is possible to be, the serelaxin is active and it's detected by the ELISA.
Great, thanks very much. I do have another question. So what prevents eosinophils from taking up nanoparticles?
So eosinophils are non-phagocytic in general, but nanoparticles are, in general, they're taken up by neutrophils, by alveolar macrophages, and the dendritic cells. And I have, as I've showed, that the CD11C marker is quite important. So so it lacks in these eosinophils, and therefore, like whatever effect that you're observing is through the CD11C positive population as well as CD206 positive population. And plus, there's another thing about the size of the nanoparticles. So we have observed previously that particles which are in the size range of 50 to 100 nanometers, so they are phagocytic. That is, they can be taken up by these antigen-presenting cells. And if it is of a lesser size, so they penetrate the nucleus, they're inflammatory. If they're more in size, so they are not taken up, they're rejected, and they end up being in the liver.
Great, thanks very much. I see you, Pam, and Dr. Fisher both active right now, if you have any questions as well.
Eleanor, would you like to go first?
Sure, thank you, Pam. You're so gracious. So my quick question to you, that was an excellent talk. Thank you for so much. So you've obviously done some preclinical work, but now you have to launch into some serious preclinical before you conclude clinical trials. So are you, do you have the facilities to be able to manufacture these under GMP? What's the pharmacodynamics? I guess my question to you, what's your next step?
Right, thank you, Eleanor. So yeah, it's actually like relaxin is a hormone, and we are like, it's already at the clinical level being trialed for like chronic heart diseases as well as acute heart failure. And so that stage is there, but we are currently looking into clinically feasible routes of administration, as this involves just intranasal. But, there is always the trouble of having like mucociliary clearance. So trying to grab the efficacy. So my next step would be to manage quite a good regime of these particles for delivery into patients. So actually, these particles are under trial for intravenous at the present moment. So if we are successful in that, we'll move towards the therapy for that and clinical trials phase 1.
Thank you. Over to Payam.
Thank you for the really great talk. And also thank you for being here it must be almost midnight where you are. So we appreciate the dedication and the flexibility to join us Sunday night. Thank you. I have a kind of comment / question and then another one. So one thing I'm thinking about is with the current pandemic, both with the amount of harsh chemicals that are being used to clean, as we know, there's a link between dermatitis, let's say, and asthma development. And with just the nature of disease in general, we're going to see a huge spike in allergic airway disease in the next 5 to 10 years. So I want to know your thoughts on that. And also, the other question was about using this sort of treatment, kind of intervention at this stage versus, I'm sure you know about a couple of other drugs such as anti-IL-4, anti-IL-13 that are trying to be a little bit higher up in the chain in terms of shutting down inflammation. So what are the benefits and advantages?
Yeah, thank you. So I'll answer your first question. So the most important concern right now is particles of a lesser size, like ultra fine ones, which is due to pollution, and pollution aggravates asthma. Other than that, you have storm asthma, you have panic asthma. So other than that, if we are just talking about allergens in the air, like we said about post-COVID effect, definitely is gonna impact a lot. And so, as I said, like on my starting slide, there's only 10% of the population which are like corticosteroid resistant, but I would still say that corticosteroids are the major players here in terms of reducing the exacerbations in an acute stage. But for long terms and for patients which are like resistant, there are other forms, like you said about IL-4, but it is also about reaching them to everyone all over the world, as well as the expenses regarding that, and having like alternate forms of therapy, which is safe and also easy to administer. So in 10 years' time, it would be necessary for us to look into like alternatives. And so like one of the vaccines that's being right now developed also uses nanoparticles. And this is going to be the next generation trend, using nanoparticles as delivery vectors for different drugs and peptides. So yes, it could be a theme. And for your next question, so if you, so sorry, can you repeat the next question?
So the second question was about using kind of higher up, earlier on in the pathway to asthma, trying to inhibit things. And actually, just to make a comment about what you said with corticosteroids, I guess corticosteroids we know can inhibit regulatory T cells and other long-term anti-inflammatory effects. So I agree with you that we need to change the course of treatment completely, because while they might treat it for a week, long-term they'll have much worse effects for the patients. Right. Yeah. So the second question was, what do you think about serelaxin versus anti-IL-4, anti-IL-13, and so on?
Yes. So that's exactly a good question. So serelaxin has been shown to have the anti-fibrotic effects, and that is the key model or the key player why we are trying to use it as an anti-remodeling drug. So not only for chronic allergic airway disease, we have showed this in cardiomyopathy and acute heart failures in the clinical trials, and other fibrotic diseases of the kidney, the liver. And in all these cases, it has been able to reverse the remodeling that is associated with the organ. So no other drugs has been shown so far, or there are like pirfenidone and enalapril, but it's serelaxin, which is a major player, which can actually take care of the remodeling effects. And therefore it could be like a next thing where like we are trying to use relaxin as a form of therapy.
Thank you so much. I really enjoyed the talk.
Thank you, Pam.
Great, thanks very much. That was an amazing talk. If there are any more questions coming in through the Q&A, they might show up for you and you can monitor those as they come. And if anyone has any more questions, feel free to add those to the Q&A as well. Thanks again for that talk. That was really great.
Thank you. Thanks, Robin.